Hello and welcome, everyone. For today's webinar, we are hosting Dr. Jerry Radich to speak about a revolutionary clinical trial for myeloid malignancies, myeloMATCH. Dr. Jerry Radich is a member of the Clinical Research Division, the Director of Molecular Oncology Lab, and the Kurt Einstein Endowed Chair at the Fred Hutchinson Cancer Research Center, and Professor of Medicine at the University of Washington School of Medicine. He is the chair of the Leukemia Translational Medicine Committee of the Southwest Oncology Group. He was the inaugural chair of the NCI and NIH Leukemia Steering Committee and the past member of the Board of Scientific Counselors of the National Institute of Health Genome Research Institute. He is the past chair of the CML Committee of the National Comprehensive Cancer Network and a member of the European Leukemia Network CML committees, and is on the scientific board for the International CML Foundation and the MAX Foundation. He currently leads the laboratory committee of the NCI AML Precision Medicine Initiative, myeloMATCH, and leads the foundation of the NIH program in measurable residual disease in AML. So a big welcome to Dr. Jerry Radich, and I will let you take the floor.
Hi, everyone. I'm Jerry from Seattle, and let's get rid of this so I can see my screen here. What we're going to be doing here today is talking about a rather huge program that we've decided to start up in the NCI called myeloMATCH, which is a cooperative agreement between all the U.S. cooperative groups and the Canadian cooperative groups, so that all newly diagnosed AML and MDS patients can be enrolled into a network of trials based on their molecular and cytogenetic profiles. So to set this up, first, a tale of two leukemias. Above is CML, below AML. And these are SEER data, and you look at the death rates here, and you can see that for CML, it was chugging along here and then suddenly dropped down dramatically. And that's because that was the introduction of tyrosine kinase inhibitor therapy. And this basically shows the power of having a molecular marker that you can both target pharmaceutically and use for monitoring disease. And so the revolution there is that now patients with CML in chronic phase in the United States have basically the life expectancy of anyone in the normal age-matched population. And from a practical standpoint, from clinical research, because the BCR-ABL test is so good that you can actually now, all the phase 3 trials for second and third generation tyrosine kinases have used BCR-ABL testing in the peripheral blood at the 1-year mark as the primary outcome measurement. So we've basically turned in CML 5 and 10-year survival trials into 1-year trials based on molecular marker. And that's really the poster child of what we like this so-called precision medicine to be able to have marker-driven therapy and marker-driven outcomes. Now, below is AML, and you can see that the death rate really hasn't changed at all since 1992, and actually it goes back farther than that if you want to. So the question is can we basically use modern molecular methods and new targeted therapies to change AML into CML? And so you've all seen these things before. This is basically looks at the spectrum of mutations and translocations in AML. And it's the good news is that most of the mutations and cytogenetic lesions in AML, maybe 20 to 50 markers at most as compared to solid tumors. So it's not quite as simple as AML or CML, but it's a lot simpler than the solid tumors. And on the top right here are circus diagrams basically showing how certain mutations congregate with other mutations. So there aren't an infinite number of mutations. And most patients with AML will have somewhere to 3 to 5 driver mutations. So it is sort of an area that you can imagine as an intermediate point in precision medicine to kind of broaden our hunt for agents and broaden our hunt for targets. Classification schemes in AML and MDS are kind of a cottage industry. Actually it's sort of like you can tell how much effective your agents are by how many classification schemes there are. It's kind of an inverse relationship. So when your drugs aren't that good, you basically try to make hay by making more and more classification systems, which makes you think like you're doing something useful for the patients when actually you really aren't, but it helps our prognosis a little bit. So obviously we can turn, figure out good and bad prognostic patients. This is the latest scheme of doing this. But the question is then, can we actually leverage this kind of knowledge into actually improving outcomes? So as mentioned, the NCI and the NIH now have a fair amount of interest in precision medicine and in AML. And so we're going to be talking about here on the left, the myeloMATCH, which is our AML precision medicine program, rapid diagnostics, which we try to get in 3 days, trials based on targets, MRD eventually is an endpoint, and we'll have MRD eraser trials. We mentioned the FNIH program in AML, which is really looking at both creating standards for next generation sequencing technologies in MRD, basically doing bake-offs into different types of technologies, and really leveraging lots of retrospective data to give the FDA an angle of how we can use MRD as a potential surrogate marker. On here on the left, this just highlights that precision medicine is a lousy term because this is precision, and that's actually what we don't want. So your byline for precision medicine could be we're reproducibly wrong, which probably isn't what you want. And here's a quote by John Maynard Keyes: "I'd rather be vaguely right than precisely wrong." So this is what I used to think about precision medicine. You would basically get a patient who's sick here, and we would generate some data, and lo and behold, we'd pick out a drug based on that data, and you'd have this, happy guy coming out. The actual nuts and bolts of getting myeloMATCH together was considerably bigger than that. I thought that the main pitfall would be making the data part, the lab part, and as I'll show you, it's only a small part of the problem in making a network like this work. This is sort of an early cartoon of how we would imagine myeloMATCH. You basically have local diagnostics, which is needed to make the diagnosis of AML or MDS. The FDA will not let us do that based on molecular lesions. You have that, and you have whether the patient's age and whether they're suited for intensive or non-intensive therapy, and get the cytogenetics and myeloid data that we get. And you churn through these gears, you'll be put into older, younger, or MDS brackets. This gear churning stuff has become, more and more symbolic as we've gone through it, because on many weeks it feels like I have been put through one of these kind of grinders in getting this trial going. But as again, the theme here is that we're going to have genetically driven protocols. They're going to be predominantly phase 2 trials as opposed to what we've usually done in the intergroup with phase 3 trials. These are going to be almost all phase 2, and they're going to be predominantly driven by MRD endpoints. At first, they're going to be driven by flow cytometry. So that flow cytometry usually will be done, at the end of consolidation, and whether you have residual disease or not kind of determines whether you stay on study or go off study. And this is sort of the tier system. And you can imagine the tier system is sort of based on disease load. So the first trials here are the tier 1 treatment trials. Those are for newly diagnosed patients. And again, we'll have baskets for older, younger diagnosis. Tier 2 trials are for those people who after tier 1 have residual disease. And so these will be so-called eraser trials that will be put up. Tier 3 will be patients who can either go from tier 1 if they're high risk, or after tier 2, that will go into transplantation or cellular therapy trials. And then lastly, tier 4 will be trials based on ultra-sensitive next-generation sequencing. So clones that you have left over after all of this. If we can find disease burden there at, let's say, 1 to 10 to the 5th to 1 to 10 to the 6th level of a VAF, then targeted therapy for that. So that's sort of the tier arrangement right now as it's set up. We've worked the network so that samples that are taken will go for local therapy to get pathology and whatever other studies you want. And then we'll send studies off to the central labs. Now, these central labs were picked by a very secretive process by the NCI contracting group, Leidos. When we discussed what kind of things we wanted to have done, We couldn't mention labs by name or companies by name in any of the process, so not to bias our own thinking. And I've accused our center from doing any work because I was kind of the head of the lab system organizing this, and I thought that'd be a bad look. But nonetheless, when the secret stuff was done, the Leidos dumped the contract on us. So there you go. And so Children's Hospital will be doing the flow. Brett was here for ages, but he's left, so the flow moved with him to Children's. Um, the molecular characterization lab at Fredericks and our lab at the Hutch split the next generation sequencing responsibilities because we work on the weekends and they don't. And then Min's group in cytogenetics, again, working 7 days a week, will do the cytogenetics with the aspiration that all this stuff will be done within 72 hours. So the essential components of putting myeloMATCH together, there's really 4 groups that we have that did sort of, independently stuff that on their own and then come back together in a larger group. One was the information technology team, which basically had to set up an information system where any of the community hospitals attached to Cooperative Group hospitals could enter patient information, sign up for trials, and then have a tracking system that follows the labs, follows the samples from center to lab and back again. So it then integrates all the lab-based into a centralized system and oversees treatment assignment algorithms and trial assignments as people go through the tiers. So the idea is that once people go into myeloMATCH, they will be tracked forever in and out of different tier assignments. So we'll get a very long longitudinal study of people, how they respond to different trials, be able to study stuff like clonal heterogeneity and its evolution, et cetera, et cetera. But the information system to set up this took quite a number of years, and it's still being tweaked some. There's an agents team, because basically what you have to do is find new drugs that aren't FDA approved. And that means finding pharma companies that want to cooperate and developing contracts. There's the laboratory team that I talked about, and we had to work with the FDA to validate these for IDEs because we're all using new drugs. And then there's a trial team, which basically has to integrate the agents and the testing and work with pharma for an acceptable design, because sometimes pharma wants the control group to be, a pretty lousy control group so their new drug looks better. And, that's often not what we might want as clinical investigators. There's always some negotiating there. This is just an example of one of the scores and scores and scores of wiring diagrams that basically shows how things go through enrolling sites, the biorepositories, the labs, back to Matchbox, which is where all the data goes back for treatment assignment, et cetera. And so this stuff all had to be put together. So it was an enormous amount of work to basically build these operating systems and test them. Obviously, I had to go do an awful lot of testing and stress testing before we ever went live. This is sort of the Ancient Genes Working Committee, and what I want to highlight here is that this really takes a long time. So down here, 200 to 228 hours of actual time from this committee for each agent brought into the program. So lots of negotiation, lots of contracting has to get done. And it still takes 60 hours of someone's time for agents that never actually get in the program. So this is a major bit of group to get just the drugs that we're going to use involved. We'll talk more about this in the future here about the initial patient assignment, which we want to do within 72 hours. And that's obviously, as you guys can imagine, quite a push. And so what happens was when people come into the system, they're enrolled into this master screening and reassessment protocol, which basically gives us the permission to grab samples, all the way from diagnosis and throughout the protocols. The actual time point once they're on therapy is obviously going to be protocol to protocol specific. But the general framework of getting their diagnostic stuff done and all the assignments is done through the master screening protocol, which every center has to have that wants to participate. And then every patient obviously has to go through that hurdle first. For patients who don't have a drug assignment or a targetable drug, which is, really going to be the majority of patients, we have this tier advancement system, which basically allows patients to be followed with whatever therapy the physician and the patient want. They can come back into the trials if they become eligible later. For instance, they may not have an eligible trial at diagnosis, but they still have residual disease after whatever therapy they got. And they might want to then go to one of our transplant protocols that is in myeloMATCH. So there's an ability to go in and flip in and out of TAP, and everyone gets tracked while they're on TAP. And the initial idea was also to get every sample during the treatment of TAP, but as we'll talk about, that tended to be too costly at this point. How we're doing the molecular testing is with Thermo Fisher's Genexus, which it used to be Ion Torrent, it's now Thermo Fisher. For those of you who don't know the nuts and bolts of this technology, this is a method that does sequencing by synthesis and by looking at changes in the pH that's made as you specifically put on one base pair to the other. So it can be thought of as the world's most expensive pH meter. This has an automated library that you can do, which the template is prepared by isothermal amplification. And again, as I just said, that basically it's done on semiconductors, which pH sensing. This can be done with parallel RT PCR for RNA and DNA, so you can run both things on the sample. And so this is the current panel as it stands right now. There are 1,600 DNA hotspots. Basically, 800 fusions can be captured. And this is sort of the panel of hotspot genes, full genes, driver genes, and expression, basically housekeeping genes, and specific genes as well. This is kind of a look at the kind of time frame. And this is if all goes perfectly well, which it never does. The library prep takes about 11, 12 hours, and then templating and sequencing takes another 6 to 8. So theoretically, you can actually go from input sample to getting stuff out in 24 hours. Often there's a little bit delay from the things, but we usually can get stuff done off the sequencer at least within 48 hours. And then there's obviously time that we have to go and check the analysis and the like. So it will cover about 93% of the variants that occur with at least 3% of the cases of AML. So a pretty good broad panel. And this really works quite well. This is data from our IDE with the FDA. This document weighed out at 1,400 pages of data. So we never want to do this again. But this was what we had to do by the FDA. So there were a number of cell lines we have as well, but we also had basically all together 163 samples that we had to do both at MoCA and ours to kind of prove that we could do it in both places. And here's basically how it highlights for SNVs, indels, both by acceptance criteria of how well the sample was processed and how often it failed. And then this is looking at the indels and SNVs and the fusions of overall sensitivity, which you can see is excellent and pretty much identical between labs. This is again the accuracy data that we did, again, over 99%, and it's pretty much acceptable for both labs. So sensitivities are great, specificities are fantastic, reproducibility, predictive agreement, and negative percentage agreements are excellent. So this is a pretty nifty system, and it works, frankly, a lot better than I ever expected it to. So if you'll wait now. This is the data from the Illumina blank experiments that we did, which found no false positives for all the variants. That's great. Illumina reporting we're going to make the limit of reporting at 5% for SNVs and indels, and a little bit lower for FLT3 ITDs. Fusions, we need to make 100 reads to call it well. The data is there, and actually it's more sensitive than this, but this is the level that for a diagnosis, we felt both lab-wise and clinically relevance would be a reasonable level. And this is the limit of detection. And again, that was done by looking at 1.15, the critical threshold. And at that level, we get about 100% call rate for a VAF of 5%. So that's fantastic. Limitations, there's a few blacklisted variants because you just don't get enough coverage for whatever reason in the sequencing. So these are the blacklisted ones right now. We just can't call those. Run failures, it looks like we're going to have about 5 to 10% run failures. We can still meet the turnaround time, if we have two machines. If one of the machines is open, we can just pop over to there. And there is some limitation with ITDs with FLT3. Now, there's a data I can talk about later that where we looked at data from a COG trial was we had capital electrophoresis is the gold standard. We sent to 4 very, very well-known next generation sequencing labs and asked what the VAF was. And some labs perform well, and some labs, the VAFs that you get by sequencing isn't anything near what you get by capital electrophoresis. So the lab-to-lab variation is pretty interesting. What we found in ours is the level of VAFs that we missed, is our very low frequency. So that's not going to be clinically relevant anyway. But one thing that is interesting is that the size of the ITDs that you can actually detect, that varies from technology to technology. For this method, it looks like we top out at about 120 base pairs, and above that, we can't pick it up. From our data on hundreds of cases of FLT3, that's going to be about 5 to 8% of patients with FLT3 you just won't catch by this technology. Reported that to the FDA. The FDA was cool with that. It's just a cost of business, but that's true of all technologies, and you may or may not know what the range is on your technology. This thing spits out a report. This is an example of it. It's actually pretty readable. This one, I guess, would be the conclusion, which is on the left there. And on the right is the paper that outlines all of this data is just come out last week. And so it's there it is in the right, Journal of Molecular Diagnostics. So you can really read the gruesome details. If you have technical questions about this, it's probably best just to email me, and I can make sure that the right technical person can address your questions, because it probably won't be me. The last bullet point here is interesting. So how we do this is once all the data is in, every day at 10 o'clock Pacific Standard Time, including Saturdays and Sundays, we meet, meaning 2 laboratorians and a couple of clinicians, to go over all the cases that are ready for assignment. And then we basically assign what treatment they're going to be on, based on the availability of the protocols and what their molecular characteristics is. We get constant stream of emails. These are things that sent us. This one just basically tells us there's a new patient being registered. These ones basically tells us on the left that the flow's been confirmed, and on the right that this person is ready to review and report. And we get constant streams telling us if we're running behind. So my email is just jammed with myeloMATCH notifications about where patients are in the process, and if any of the labs is running behind for some reason. And this is just a life history of a sample. So Wednesdays, the samples collected at 11 a.m. and the path reports were loaded into the system. We received the sample on Thursday morning. Saturday, cyto and FISH is done and put in. Sunday flow is uploaded. You can see that's Sunday at 12:20 a.m. So Brett Wood apparently never sleeps. Next generation reported out, and end of the day at less than 72 hours on Sunday. the bioinformatics teams reviews and assigns the report. So this can be done. Now, one of the things that we want to do with this eventually is we want to move away from flow cytometry to some next generation sequencing techniques for both outlining mutations that will go in the tier 4 trial and for looking at MRD as a better mousetrap compared to flow. So this is a paper that we published that we did here with TwinStrand, which was a spin out of UW that I've been working with for years. Sent a number of my lab people over there to start running that place. And they use duplex sequencing methodology. So for those who haven't done it, it's an error-corrected method where you basically make libraries off both strands, and you don't make a mutation call unless the right complementary base pair is mutated in the exact same place. And so that gets your theoretical detection rate at about 1 to 10 to the minus seventh. Clinically, you really don't get anywhere near that, well, just because of the input amount of material, but it has exquisite possibilities. And on the left here, this is looking at just a meta-analysis of MRD in AML done by Nick Short, showing that MRD negative patients obviously do better than MRD positive, but still about 40% of patients who are MRD negative still relapse and die. So it's not the best of tests. And what the FDA would like for using MRD as a surrogate is, you can imagine grabbing those two curves and pulling them apart. They would like the MRD negative to be basically near 100% and the MRD positive to be basically down to 0% of survival. So that's what a better test will hopefully do. And this is an example again of the duplex sequencing. On the right is the most illustrative example. So this is looking at a patient with the 315I mutation in CML. And this is basically sequencing across the whole ABL kinase domain. And you can see if you go on the VAFs there, once you get about to 0.5%, everyone's positive, right? So these are all errors just made by making libraries and stuff. It's especially bad when you make it off of RNA and have to reverse transcript it into DNA. And on the bottom is the error corrected, where you only see the 359 mutation because you get rid of all the background. And so the question was whether we could do that in AML. And this was done looking at patients from SWOG, 67 paired patients at diagnosis and quote unquote CR, where they've had Brentwood do the flow sequencing, which is probably the best lab in the country. And on the right, the illustrative is comparing the flow negatives and flow positives to the next generation sequencing positives and negatives. You can see in red about 24% of patients are misclassified flow negative and are double strand positive. And when you look at these patients, they do a lot worse. So on the left is probably the best way to look at it. The flow down below, the odds ratio of either time to relapse-free survival, overall survival is about 2.5. That's what the meta-analysis showed that I just showed you anyway. So it's in keeping with that. But if you look at, we basically, since this was exploratory data, we looked at various ways of calling mutations by the next generation sequencing. And if you look at the best method here, which is you say a non-DTA associated mutation with a VAF of 0.1% and NPM a little bit more sensitive, you can see that odds ratio for time to relapse-free survival, and overall survival is way better than in flow. So we're hoping that we'll use some kind of duplex sequencing technology to bring in and kind of sub out flow and be able to use that in some kind of regulatory fashion. So negotiations with the FDA are ongoing. So protocol development, because you have to get protocols to go with these things. We now have 5 protocols that are going. We have one for older and less fit AML, FLT3 positive AMLs, young ELN high-risk patients, and younger ELN intermediate-risk patients. And we have about 10 protocols in the wings. The IDH2 protocols for MDS and older age patients have just been approved. They're online. And we have these various eraser trials, CBF trials, IDH1 trials, et cetera. Here are all ongoing and should be you know, up and running here very soon. So we're hoping that at steady state, we'll have about 10 trials going at all time. Now, let's talk about unanticipated issues here, because this is where things have gotten very interesting. When we started myeloMATCH, we didn't know what accrual would be like because, on one hand, there had been, with the pandemic and the like, there had been no active AML trials in the intergroup for quite a while. And as you guys all know, the pandemic really, you know, put a squeeze on staffing and the like. And so we didn't know whether people would basically just decide they didn't want to get in clinical trials anymore, or on the flip side, if there would be some kind of pent-up demand. So when we got the money to do this, we anticipated that we would have 500 patients accrued a year. And so that's the burn rate line in teal there. And then here's the actual line. We started myeloMATCH in basically a year ago, and we had very flat accrual because it takes a while for people to, you know, bring the protocols up to speed or just online. It usually takes a month or two just to get through all the hoops. So we started to see this climb, and then we started to see this spectacular explosion right here, so that we were seeing up to 25 cases a week coming in, and we were online to do almost 1,000 cases. So basically doubling the number of patients that we'd expected to. As you might imagine, that created two stresses. One stress to the budget, because as things are all aligned, we can't get another pot of money until the 1 year cycle is over. And obviously put a fair amount of stress on the labs itself, because, you know, to get these things in 72 hours were really a lot of overtime and stuff to get done. This is the assessment of the first 350 patients, though. And I just, to the arrows, what you want to look at is the average hours until cyto results are out were 70 hours. The percentage of time that cyto was done in less than 72 hours was 76% of the time, which is what our goal was. For next generation sequence, the average time coming out is 52 hours, and about 82% of people, we got the results out less than 72 hours. That's all great. But we had to change something in activity. Right now, we have almost 500 patients enrolled at 277 centers, but we had to do something to kind of put a slow down the tap here because we were just like burning, you know, people and budget. So one thing we're doing now is we aren't doing routine TAP assessments. So when people who are on this, you know, standard practice protocols, we're not getting routine reassessments banked and done in our labs until we have the tier 2 and 3 trials up and going. And so then they can reassess to go into those trials. And so we'll start them up then. We aren't going to be doing master screening protocol testing until the centers actually have the protocols relevant online, because that's been a problem because people would, you know, think that their protocols, the treatment protocols would be ready to go and they would get the screening done and we'd work them through, and lo and behold, they're eligible, but they're not eligible for the trial yet because the trial isn't up and running yet. So that was probably an oversight that we've rectified. And then we've put a slot system in place, so that now at 8 o'clock Pacific time every morning, if you have a patient coming in, you can go online and enroll to have them take up a slot to be guaranteed a enrollment in the MSRP. Those are usually anywhere from 3 to 5 slots a day are opened up. We're trying to keep it at 15 patients total a week. And those slots, Megan Others, who runs it, has been telling me go in 15 seconds. So people are hovering above their keyboards, waiting for 8:00:01 to hit and to register the patients. And lastly, what I'll talk about next is to ease some of the workload and cost. We're wondering whether our next generation sequencing panel of the fusion genes might be able to take the place of FISH. So here's the problem with the enrollment we talked about right now in this pieces of pie. The circles are the enrollments, and we're only enrolling about 10% of patients are actually getting onto a trial. So why such a low assignment? Not enough protocols open yet. The MSRP open, but not the protocols open. Some of our centers were kind of using the MSRP as a way to get free testing, which I understand, but that's really not the use. So we're trying to limit that. And then in retrospect, most of the patients are not going to have a marker, right? Because we just don't have that many drugs yet. So we've now have a marker negative trial with some novel agents coming online, but realistically, that probably should have been the first thing that we did, is have a marker negative trial and have that open, because if we did that, the accrual onto some type of clinical trial would probably be about 70%. So, we have many classification systems. There's no classification system that isn't better than the Sergio Leone good, bad, and ugly classification system, which almost everything in the world fits into that. So the good is accrual is great, turnaround time's hitting our mark. The bad is so far we only have 5 protocols up, others coming. And so the vast majority of patients are going on tap. And the ugly was that the burn rate's just too high. And we don't have any tier 4 assay right now because, as you might have known, Twins strand got sold for scraps by their investors. And so we're trying to find, probably bring up in our lab, the duplex sequencing. So here's some data that's sort of hot off the presses. This is from Min Fang and DJ Jawani at MoCA. Min Fang from here. And we've generated data from the myeloMATCH, and we're hoping to go to the FDA to change the IDE to allow us to use the myeloMATCH assay for fusions instead of FISH. So this is kind of how things are done right now. The patients are enrolled. You get the assay, you get the karyotype, you get a big FISH panel, and then you get pushed on EFL quantifications based on the mutations and the FISH and cytogenetic panel. So if you look at our first 400 patients, this is what the karyotype shows, and looking at fusions. So I'm just showing the fusions here because that's what our panel can pick up. So this is kind of the distribution of the fusions. You can see that it's very similar to the FISH fusions, and I'll show a more direct comparison here in a minute. And comparison with the myeloMATCH fusion assay. And so the question is, how do these compare, and can the fusion panel replace FISH? So this is how they look. If you look at mutations, of FISH and karyotype, and you see they line up pretty darn well. The big picture here is that the Genomics assay doesn't detect the inversion 3s, the GATA2 MECOM rearrangements, because the lack of amiable fusion that's in the panel there. So that you're just not going to catch the inversion 3s as the panel set up now. And the karyotype and FISH assays tend not to detect the KMTA, PTA often, and often that's in these samples just because the level that it's there in these samples, and the fusion panel by the Genomics assay is just more sensitive. If you compare karyotype and FISH head to head, just the fusions, you can see the overall agreement rate's 98%. You get a few that are missed, one way or the other, and we'll discuss those in a moment. If you compare the Genomics and the FISH, the comparison is really quite good, 99%. And the discordance is those that have you a positive fusion and a negative FISH. And that seems to be just basically that some of these subclonal populations were just too infrequent to be picking up by FISH, whereas you could by the Genomics assay. And this is, I'm not going to go through all of this. This is just looking at why the discrepant results between the karyotype and FISH. These seem to be mostly false negatives by the karyotype. And the reason below in the highlighted box, most of these basically cryptic translations that FISH is just better at catching than cytogenetics. Differences between the karyotype and the MMA results. Again, these tend to be false negatives from the karyotype for kind of the same reasons, that basically a sensitivity issue. and differences between FISH and to the Genexus assay. These were pretty much all because of BCR-ABL. There was this one case that had a fairly high amount of BCR-ABL that the FISH didn't get. That could be real, that could be not real. As you know, most of us harbor some BCR-ABL if we look hard enough. And then here there are a few where you just pick up very, very low levels of BCR-ABL that just seems to be background noise in this assay. So altogether, pretty darn good results. So in conclusion, you can read this as well as I am. It looks like we think that we can convince the FDA to abandon FISH, and how this would be done is that in this modified assay, basically we'd use the Genexus assay to look for the fusions, but we'd have a inversion FISH 3 fusion FISH assay still going. Also, we'll have a P53 FISH assay going. So it basically reduced the FISH load by probably about 75%. So that should really kind of help workload and cost, hopefully. So my final slides, we have some time for questions. This is sort of what we want, right? We want to be able to diagnosis is to look for mutations that will drive prognosis and drug choice. In the middle here, we want to look at MRD so that we can change drug if we need to, change therapy, build MRD eraser trials, and maybe use these for regulatory endpoint. And then, oops, sorry. And then what we want, the wish list, is we want fast, reliable, sensitive, flexible, and cheap assays. But one of my favorite musicians is Tom Waits, and this is the director Jim Jarmusch telling Tom Waits, fast, cheap, and good, pick two. If it's fast and cheap, it won't be good. If it's cheap and good, it won't be fast. If it's fast and good, it won't be cheap. Fast, cheap, and good, pick two words to live by. And with that, I thank you for your attention, and I'd be happy to entertain any questions.
Thank you for the wonderful talk. So I'm going to start asking the question. The first question is, what is your approach to filter germline from somatic variants, especially in genes associated with coronal hematopoiesis?
Excuse me, what's the question again? I didn't get that. Sorry.
What is your approach to filter germline from somatic variants?
Yeah. So we do not routinely do that. We have samples collected for a research basis. But at diagnosis, we're making the call of AML based on pathology locally. We can't do it by molecular. And so in that case, we are basically just not going into the germline issue.
Thank you. The second question is, does this assay mainly target hotspots? And
if you go back to my panel, which I'm sure you guys are going to get the slides on, about half of our hotspots and about half of the mutations are full gene.
And do you report the VUSs on this as such on this assay?
Yeah, we will report them. No one knows what to do them, but we will report them.
And what is the burden of variant classification, if any, in the analysis pipeline?
So I think you mean the verification classification system is actually done by Thermo Fisher. So it's an automated process using kind of an amalgam of many of the classification systems out there. But then we are creating our own sort of library of this. And so even though these mutations are called by the automated system by the machine, our variant callers are going through all the data just to confirm them.
Thank you. And there's a question that was actually my question too. Could you comment on how to track MRD status for AML with defining gene fusions?
Oh boy, that's a great question, right? So the number of actual fusions that are out there that are FDA approved and everything, it's just a handful of them. So you can't really do most of those, right? But if you're required to do that, in general, the fusions are more sensitive than flow. As you know, flow is very hard to standardize and varies markedly from lab to lab. We did something with with the FNIH years ago where we sent the same exact sample out to 4 commercial labs and academic labs, and it was almost a shotgun shot a blast of what they said, right? Um, so my feeling is if you have a good homegrown fusion assay, with the, good standards and all that, I think that that's perfectly fine to fall and probably better than, most cases of flow. The only possible issue with that is, we see cases in AML that have no residual disease by flow, but will be positive by either, sometimes even FISH or any molecular lesion, either a fusion or mutation analysis by a sensitive method. And we say on one hand, maybe that's just, the sensitivity issue. But on the other hand, you'll get patients if you follow who will have tractable MRD and not relapse. And so what probably happens in a subset, and single cell work has shown this, is that since these are stem cells diseases, you will often get these lesions in lymphoid cells. And so you'll be able to track the fusion or the mutation in a population of lymphoid cells that will be positive, but are never going to expand or relapse. And so that's why I think some of these technologies are complementary.
Interesting. Thank you. And then I think another question is regarding the controls. How do you include the controls like positive, negative, and NTCs in each clinical trial NGS run?
Yeah, so that's a really good question. So we use sort of standard, cell line horizon controls in runs. But I think where the field is going to go, and this is the work that my part of the work on the FNIH stuff, is there's a number of groups now that are going with spike-in controls. And I think in the future, spike-in controls, we use spike-in controls to each sample, especially if you're getting into MRD issues. That's probably the way to go. And those are being developed now. There's a group called Acute Genomics, who's made a panel of spike-in controls. And we've shown in our lab, and this will be published soon, that you can spike in that VAF you get at very low levels without the spike-in controls is the same as the spike-ins. So the spike-ins aren't competing for the target. And it really, if you're getting down to ranges where you're using an MRD like 1 in 1,000, 1 in 10,000, it really tightens up the lower edge to know that, because you know exactly how much spike-in control you should read out on the other end, right? And so you can mathematically compensate for that. So I think that's probably the way to go. And I kind of prefer doing one lane of controls in every run because, as opposed to the periodic checking, because these machines are very finicky.
That's true. So I think there's another question now. So it says, if you see cytogenetic abnormalities that are clonal but does not include fusion genes, how do you classify it?
So clonal but fusion. So those are just, your two typical cytogenetic abnormalities, right? So the cytogenetics there would basically use that for the ELN guidelines, and that drives one of the classifications between high risk and low risk. That's still almost always being driven by classical cytogenetics.
Thank you. I have a question if there's no other questions. So one of the things that I always wonder, so when they are pulling the aspirate, so we have the first pull, and then we have the second pull. So we assign it for the MRD and the flow
Oh,
cytometry. Yeah. So I was wondering what you guys require. Like, is there any requirement, and is there causing any variability between samples, and does it matter to you guys?
This is a huge thing that people don't think about. So thank you for thinking about it. So one of the issues is we try to get flow as being our first or second pull. You usually can't get it. Usually the pathologists like the first one, this the second one. But it brings to the point of that all of these studies where you're comparing flow and some other technology, they're usually not on the same sample. usually on different pools, right? And so by the time you get to the next generation sequencing stuff, that's usually like third pool, fourth pool. You're just getting peripheral blood at that point, right?
Yes.
And so like in the study that I showed where next generation sequencing looked better than flow, I bet that if you do it off the same sample, it'd be a lot better. And so what we're doing in myeloMATCH is that all the samples that Brad Wood's getting for flow, he does a flow on an aliquot of it, but then we store the rest of it. So all of our comparison studies are going to be on the exact same material that flow is done on. And I think that's the only way that you can really, figure this out. But I am hoping that actually with good enough, sensitive enough testing, I think we're going to be able to move soon to like what we did in CML and not do bone marrows anymore. I think that in the near future, all MRD testing in AML will be able to be done on peripheral blood.
Great, thank you. So there's another question. What is the failure rate due to poor sampling or shipping? And by dropping fish, are you concerned about breakpoints not covered by NGS, or are you expecting karyotype to pick up?
Yeah, we're expecting the clonotype to pick that up, right? And what we looked right now is the fish and the thing I showed with the fish and the fusion panel are working spectacularly well, way better than I would have. Now, the failure rate, that's something else, right? So there are two parts of the failure rate. There is the sample getting lost in transit or being here, taking too much time. That's actually relatively rare. I mean, I would say that's probably sample issue problem with the actual sample is certainly way less than 10%. Now, samples coming with the right documentation and forms and stuff, that's a whole different thing. And it's stunning. And it's mostly, I think, because people are out of the practice, right, of doing this. So we'll get samples without any documentation of forms. We actually sometimes get forms without samples. And especially at the beginning, it's better now, but especially at the beginning, so much of our tech time and actually my time, because if we got too many of them, I would make the calls to the local centers. But we were spending hours every day calling up to get in more information about the sample, dates were wrong, all of that stuff. We were promised at the beginning that this would all be automated with barcodes. It's not. We
Hopefully.
sold a bill of goods on that, but they're trying to get that up. But that will hopefully be the answer. But right now, still way too much of the time in the lab is spent tracking down samples and stuff. And it's not the sample quality issue. It's all the documentation with the samples.
Great, thank you so much. Can I ask another question? Maybe it is too early to ask this, but does myeloMATCH affect the time for the transplant?
Yeah, it's too early to say that. It's too early to say that. I suspect it will, because I think what happens in AML is, especially people with MRD, we end up fooling around with them too much to try to make someone MRD negative. And the issue there is that people in their heads, it's a different biology from someone who goes to MRD negative than to someone who's MRD positive, and then you kept beating and beating and beating on them, and they finally get MRD negative, right?
Yes.
I think most of the time we're just treating ourselves when we do that. So what we're hoping is that when people get, through therapy and consolidation, if they're MRD positive, if they've got a transplant option, they get a transplant. And nowadays, if you think about, many centers don't have age restriction on transplant anymore, right? And we can find donors for almost everyone, matches, unrelated donors, haplos, and umbilical cord. You can find a donor for everyone. So realistically, most patients who we treat should be transplant candidates.
Great. You know,
And thank you so much with that. I really am happy that we shared this last webinar for the season, ending with such a great topic. And I really do appreciate your talk, and some of your thoughts are very interesting, especially about not following up with a bone marrow, potentially following up with peripheral blood. So again, a big thanks
Right,
to
thank
Dr.
you.
Gerald Radich for giving us a talk on myeloMATCH. Just a quick reminder that the CGC 2025 annual meeting, regular registration ends on July 15th. It's in Houston this year, August 3rd through the 6th. Hope to see everyone there. And then additionally, if you have an interesting case, remember that the CGC Genomic Case Series is always looking for those. So any fellows or residents or techs want to present, they can, and it's a CME for a giving conference for an international community.